If oxidative phosphorylation feels like a blur of complexes and proton gradients, you're not alone, and it's one of the most tested metabolism topics on the MCAT. But what if you could see the electron transport chain as a simple logical flow of energy instead of a memorization nightmare? In this video, we'll break down each complex, track how electrons power proton pumping, and connect it all to ATP production, so you can actually understand and remember how it works on test day. The electron transport chain transfers electrons from NADH and FADH2 through protein complexes to oxygen, generating a proton gradient. This process occurs in the inner mitochondrial membrane, where four main complexes, complex 1, 2, 3, and IV, work together to pass electrons step by step. NADH donates electrons to complex 1, while FADH2 donates to complex 2, which is why NADH ultimately produces more ATP. As electrons move through complexes 1, 3, and IV, energy is released and used to pump protons from the mitochondrial matrix into the intermembrane space. This creates both a chemical gradient and an electrical gradient, collectively known as the proton motive force. Oxygen serves as the final electron acceptor at complex 4, combining with electrons and protons to form water, which is essential because without oxygen, the entire chain backs up. A helpful mnemonic is I, 3, IV, pump protons, 2, does not. On the MCAT, you are often asked to predict what happens if one complex is inhibited. For example, blocking complex 4 would prevent oxygen from accepting electrons, halting the entire chain, and stopping ATP production. This concept connects directly to cellular respiration, redox reactions, and the importance of oxygen in aerobic metabolism. The proton gradient generated by the electron transport chain drives ATP synthesis through ATP synthase in oxidative phosphorylation. Once protons accumulate in the intermembrane space, they naturally want to flow back into the matrix due to both concentration and charge differences. ATP synthase, sometimes called complex V, acts like a molecular turbine that allows protons to flow back down their gradient. As protons pass through ATP synthase, the enzyme rotates and catalyzes the conversion of ADP and inorganic phosphate into ATP. This process is called chemiosmosis, emphasizing that ATP production is powered by a chemical gradient. A useful way to visualize this is like water behind a dam when released. The flowing water spins a turbine to generate electricity, just as protons power ATP synthase. On the MCAT, a common question is what happens if the proton gradient is disrupted. For instance, if the membrane becomes leaky, ATP production decreases even if electron transport continues. This concept links to thermodynamics, particularly the coupling of exergonic electron transfer with endergonic ATP synthesis, and highlights why the integrity of the inner mitochondrial membrane is crucial. NADH and FADH2 yield different amounts of ATP because they enter the electron transport chain at different points. NADH donates electrons to complex 1, which contributes to proton pumping, while FADH2 donates electrons directly to complex 2, bypassing complex 1 entirely. Because complex 2 does not pump protons, electrons from FADH2 result in fewer protons being moved across the membrane, and therefore less ATP generated overall. Typically, NADH produces about 2.5 ATP, while FADH2 produces about 1.5 ATP. A quick mnemonic is NADH equals more ATP, FADH2 equals fewer ATP, because NADH starts earlier in the chain. On the MCAT, you may be asked to compare energy yields from different metabolic pathways, such as glycolysis versus the citric acid cycle, and understanding where NADH and FADH2 are produced becomes essential. This also connects to the broader concept of redox carriers. and how their energy content determines their contribution to ATP synthesis. Oxygen is the final electron acceptor in the electron transport chain, making it essential for aerobic ATP production. At complex 4, electrons are transferred to oxygen, which then combines with protons to form water. This step is critical because it prevents electrons from accumulating in the chain. If oxygen is absent, such as during hypoxia or cyanide poisoning, the electron transport chain stops because there is nowhere for electrons to go. As a result, NADH cannot be oxidized back to NAD+, which halts the citric acid cycle and forces cells to rely on anaerobic pathways like fermentation. A helpful way to remember this is oxygen keeps the chain moving. On the MCAT, you might see questions about what happens during oxygen deprivation or toxin exposure. And the key idea is that ATP production drops dramatically due to ETC shutdown. This concept also ties into physiology topics like muscle fatigue and lactic acid production when oxygen supply is limited. Uncoupling disrupts the link between electron transport and ATP synthesis by dissipating the proton gradient. In normal oxidative phosphorylation, electron transport and ATP production are tightly coupled through the proton gradient. However, uncoupling agents such as 2,4-dinitrophenol or naturally occurring thermogenin in brown adipose tissue allow protons to flow back into the matrix without passing through ATP synthase. This means the energy from the gradient is released as heat instead of being used to make ATP. As a result, electron transport may continue at a high rate, but ATP production decreases. A useful way to think about this is like poking holes in the dam. The water still flows, but it no longer efficiently powers the turbine. On the MCAT, you may be asked to predict outcomes of uncoupling, such as increased oxygen consumption but decreased ATP yield. This concept connects to metabolism regulation, thermogenesis, and even weight loss drugs that have historically targeted uncoupling mechanisms.